Hi, everybody. Welcome to Tuesday. In today's lesson, we are going to get right in it. We are going to go through slides 61 through 76. So it might seem like, holy smokes, we got a lot to go through, but there's a lot of like little changes in each of the slides. So it'll be just fine, hopefully. Okay, so we've been working on doing one trait per Punnett square. We called that a monohybrid cross, whether it was for eye color, hair color, the pea flower color, whatever it may be, or blood types, or any of the sex link stuff like with color blindness or and whatnot. Now we're going to take it a step up and make it even harder, but actually it's not really too hard. Um, and we're going to do two traits for the Punnett square. Two traits, we call that a dihybrid Punnett square. Um, Sometimes it's important to track two traits at one time. Um, when we're talking peas, and I just went too far. Um, peas, I don't know about you, but I like it when they're green and when they're nice and round. Some of them have the little dimples on them, but really that's okay. That's just a genetic difference between them. And so, like I said, we can make a dihybrid cross Punnett square. So it's kind of like a Punnett square on steroids. You know, the monohybrid, it's just four squares. Guess what this one could potentially be? What's 4 times 4? 16, a 16 square Punnett square. Yep, we're going there. All right. So when Mendel was doing some of his studies, some of the conclusions that he made, one of them is called the law of independent assortment. It says when characteristics are inherited, they do so independently. It's not like, it's like, okay, this one's automatically going to go to mom, or this trait's going to automatically go to dad, or it's going to automatically just split a certain way. It's all random. It's all assorted independently. That's why they're not connected to one another. Okay. One of the reasons why there's so many genetic differences in all of us and why you are going to be the only you that ever lives. Okay, so peas can be round or wrinkled, peas can be yellow or green. So those would be the two traits. How do you find the different combinations? How will you figure out the different combinations that could be given in the sperm or egg and making the peas? How will you figure that out? And then what's the probability of having round and green, round and yellow, wrinkled and green, wrinkled and yellow? We can figure that out doing a dihybrid Punnett square. You're going to hear me say this thing foil. We are going to use, now that dumb thing at the bottom came up, foil. And some of you may be familiar with this and what it means from math class. No, I'm not talking aluminum foil, but foil actually stands for first, outer, inner, last. That's how we find all of the possible gamete combinations. So the possibilities would be round and green, round and yellow, wrinkled green, wrinkled yellow. Okay, and what does that look like? How do we foil? Step one, who are the parents and what is their genotype? So round we know is dominant over wrinkled, green is dominant over yellow. So it says one pea plant is heterozygous for both traits. Oh, that stupid thing keeps coming up on the bottom. And so what's the genotype of that parent? If they're heterozygous, I'm actually going to flip this over and look at the board because I'm going to be writing some things. This seems like it's really bright. Hang on. To adjust the light. Okay, hopefully that's better. So if this parent is heterozygous, I'm going to use R's and G's. So heterozygous, I know for R's, it's big R, little r, and then we use G's, big G, little g. So the R's are going to represent round or wrinkled, and then the G's are going to represent green or yellow. Okay. I think I have too much light still. Okay, hopefully that's better. And so that is the genotype of that parent. Big R, little r, big G, little g. And then for this, you don't necessarily have to do it alphabetically. Like the G's would go first over the R's. You can if you want, but you just need to keep the G's together and the R's together. The R stand for round or wrinkled. Okay, the G stands for green or yellow. We said that. And so now to find the possible gamete combinations of the alleles for that this dad could pass on, we're going to use FOIL. First, outer, inner, last. So when I say for foiling this, the first combination would be the first two of the big letters. So that would be big R, big G. The two outer ones, big R, little g. The inner ones, little r, big G. And then the last combination, little r, little g. So those are the possible gamete combinations that this parent could give. And I think that's what this all just goes to. So that would be the first two. The outer are the outer two letters. The inner are the inner two letters. And on this one, I know the capital is like would end up being written second in dihybrid crosses. That's okay. And then the last two are the last two that are left, little r, little g. Okay, let's try one more. The other parent, it says, is homozygous round and heterozygous for green. So when I see homozygous for round, I know round is dominant over wrinkled. And if they're homozygous for round, there's only one possibility that that one could be. So it's big R, big R. And then heterozygous for green color, big G, little g. Okay. And then we're going to foil this one too. Let's see. What? Write the mom's genotype. Okay, we got that. And then we're going to foil this one. So if we do first, so it's big R, big G, outer, big R, little g, inner, big R, big G, and then last, big R, little g. It's a G right there. So those would be the gamete combinations for the other parent. I think this was for the mom. Okay. Now, today we're just going to take it one step. I just wanted to show you all how to do the FOIL method because then tomorrow is when we're going to actually plug this into a Punnett square and see what all happens with this. So watch this video. Gotta love the Amoeba Sisters. They're really good with explaining things, and you're going to kind of see what we're going to do tomorrow too. But don't worry, I will help you along the way. And then you just, oopsies, have a formative that you got to go do. So hopefully you took notes on this. Rewatch it if you need to. Go back and make your notes on foiling. If you have questions, please let me know. But tomorrow is when we will plug it into the dihybrid, the big Punnett square. You're gonna love it. Hope everybody's doing good, and we'll see you soon.
Hi.
Bye.